Hello, I'm Patrick Hwu, President of the Society for the Immunotherapy of Cancer, or SITC, as well as President and CEO of the Moffitt Cancer Center in Tampa, Florida. And we're here today. We're honored to be with Dr. Ugur Sahin, one of the world's foremost experts in immunology and cancer research. He was a co-founder and CEO of BioNTech, and also the developer of the first approved vaccine for COVID. So we're really pleased to be with you, Ugur. Thanks so much for spending the time today with us.
Thank you, Patrick. It's a great pleasure.
Tell us a little bit about how you got into cancer research, medical research in the first place.
I'm a physician and tumor immunologist, and I did parallel to my education as a physician research, and at a certain time point I understood that we need to do more than doing research in the lab, but also try to bring these ideas and new therapies into the clinical practice. And therefore I decided to become a translational researcher. And on this way, I also decided that we need to enable this extremely expensive treatments and development in a more proper way. And I became a co-founder of a biotechnology company.
Wonderful. Why were you drawn to immunology?
It's the most fascinating research topic. I really love immunology. It is on the one side the complexity and all the wonderful mechanisms, and on the other side, I always believe in the power of our immune system to affect disease, not only in cancer but in a number of diseases, infectious diseases, autoimmunity, and even inflammatory diseases, and I always believe in the opportunity to use the immune system also for personalized treatment. So it's a fascinating topic.
Wonderful. We're all really proud that you're a member of SITC, that you started all of these vaccines in cancer research. I remember speaking to you at a KIMT meeting a few years ago in Mainz, and you presented your really impressive results where you were immunizing against melanoma using the RNA-based vaccines and getting really high levels of T cells in the circulation from these vaccines. Can you tell us why you centered and focused on RNA-based vaccines for your research and practice?
Yeah, so we early on realized that cancer is an extremely heterogeneous disease, not only at the genetic level, but also on the level of antigens, of tumor antigens. And the key element in cancer and developing cancer vaccines is understanding what kind of antigens can be used. And we know that there are at least two classes of tumor antigens, which these are the shared non-mutated tumor antigens and the mutated ones. And both are relatively heterogeneous. So we were looking for a technology which could allow us to deal with this inter-individual variability. And the other thing was we were interested really in using powerful T-cell responses. And I was fascinated with my very first experiment more than 25 years ago when I transfected mRNA into dendritic cells and saw how powerful mRNA could be to deliver antigens into dendritic cells, which are the prime stimulators of immune responses.
Yeah, very impressive results. When did you get the idea that you could focus your cancer research onto infectious diseases like COVID?
Yeah, this is indeed an interesting situation. So we actually focused in the last years in developing personalized neoantigen vaccines, and the idea of a personalized neoantigen vaccine is that you get the genetic information of the tumor, the mutation profile, and then you make a vaccine on demand. And we implemented a number of processes allowing us to make a vaccine extremely fast, because of course the patient is waiting for the vaccine, and the tailoring and development of the vaccine should take only a few weeks. So we had everything in place. We had the ability to make a vaccine according to the genetic sequence, and we could do it fast. And when we understood that this outbreak in China really could become a global pandemic, we feel the obligation to use our technology to develop a vaccine as soon as possible.
Well, it's quite impressive what you did. But what you were doing with cancer research was actually a lot harder. You were making a personalized vaccine for each individual patient. I mean, how long did that take from the patient hitting the door to getting them the vaccine if you're making a personalized one based on their antigens?
We started this approach so the first clinical trial was started in 2014, and the turnaround time when we started that was about 3 to 5 months, because it was required to get the tumor in, to do the sequencing, to prioritize the mutations, to make the vaccine, to have the quality steps, and so on. And everything was done according to GMP requirements. And everything was done by people who were pipetting and doing that. In the meantime, we have established electronic documentation, and we have implemented automation, semi-automation. And the process is now done to less than 6 weeks. So for some patients, we are able to deliver even in less than 4 weeks. So it just shows the progress if you implement these processes and ensure that the time for every step is minimized. Then this can be done, including sequencing and vaccine manufacturing in less than 4 weeks, which is in line with the usual time for decision making and treatment of a patient with any other type of therapy.
Well, that's so impressive that you can make a personalized vaccine in 4 to 6 weeks. That's really very impressive. So I guess it seems that doing a COVID vaccine is almost trivially easy compared to that. But still, how did you get this to FDA approval in less than a year? I mean, even really the experts everywhere were saying it's going to take 3 to 5 years to get a vaccine initially, and here you did it in 11 months. How did you get that done?
So when we started our project, we expected that this really will become a global pandemic, and our goal from the very beginning was to develop a vaccine and make it available in less than 1 year. We named our project Lightspeed, and the idea behind this designation was that we should not lose any time. So we did not have time to waste. And what we did is we really implemented a 24/7 research program. When we started, we did not know what is the best vaccine candidate, so we started with 20 vaccine candidates. We did the full preclinical testing, immunogenicity evaluation of this candidate. We did even a GLP toxicology study, yes, because this was a prerequisite from the German authority, the Paul-Ehrlich Institute, to evaluate that. We did the GMP manufacturing, and when we started the program, just a few days later, we contacted the German authority and asked whether we could have a scientific advice meeting. And we visited them and showed them the plan and asked whether they would be able to do a rolling review. So that means every time when we have additional information available, that we submit this information so that they could allow us to start the clinical trial in shortest time. And this happened. So we submitted our documents on April 20, and 3 days later, we had the approval to start the clinical trial in Germany. Moreover, we did a partnership with Pfizer, and Pfizer took care that the same trial was copied, and they started the trial also in the US with the FDA. And our plan was to have a seamless approach to get all the immunogenicity data from the phase one. And we understood that one of the vaccines or two vaccines provided really strong antibody and T-cell responses. Made the decision on July 24 for one of the candidates, and on July 27, the phase 3 clinical trial started. So actually, the authorities, the regulators, collaborated with us in an extremely efficient way. We were in touch with them every second day, and only by this transparency, by the collaboration, by investing a 24/7 approach, so that every day, night and day, the project could progress. We were able to come up with a vaccine in a time frame in less than 11 months.
Having done a number of IND trials and T-cell therapy and vaccines, I'm just so impressed that you got this to people within 11 months. It's just really outstanding. It's really nice, though, that you had a company also, BioNTech, that was already doing this in cancer research. I think that really helped it along. So tell us a little bit about starting BioNTech. You were a professor at a university. You had a bunch of postdocs and graduate students, like many of us. What made you decide to start a company?
Yeah, so I'm still professor at the Johannes Gutenberg University in Mainz, and so we did research, and at the early phase of my postdoc time, we started collaborations also with pharmaceutical companies. So hoping that the pharmaceutical companies will catch up what we do, and we could work together, and so that the company could continue with our ideas. But it turned out that this does not work. So after 2 or 3 years, even though the projects were successful, the companies decided to stop that because of strategic reasons. And at a certain time point, I understood, or we understood, that if you really want to bring something to patients, and not only to patients, but also to people, that means to get an approved drug, you have to start a company. So we started that just being interested and having the vision to develop medicines. And we tried to do it in a way that we still continued our research. So the way we wanted to do, we were not interested in business. We were really interested in translation. And the company that we have started, BioNTech, is more or less as a big research organization with of course sufficient funding, allowing us to continue in the best possible way, to combine in the best possible way biotechnology and science. Yeah? And it turned out that this works, because we have been able by doing science, bringing innovation to patients, to progress not only the science, but also to come up with an organization which can make a difference. And yeah, this is encouraging. It just shows that we need a better interaction, yeah, a seamless interaction between research development, and following the ideas that also other companies accomplished, for example, in the CAR T cell field.
So it gave you more control of the translational process. So it really helped you, allowed you to have more control and make that more effective and quicker too. I'm sure if you didn't have your own company, you wouldn't have done it in 11 months. Really quite impressive. So now, many lives will be saved because of this COVID-19 vaccine that you've created. How does that make you feel?
It is, of course, a huge privilege to be able, as a researcher, to make a difference and to help people. And this is fantastic. It is encouraging. It's motivating, and it just motivates us to continue with what we do, believing in technology, believing in science, and being able to make a difference is the greatest privilege that you can have as a researcher.
It's wonderful. Now we have the whole world saying we want it now. So tell us a little about the production capabilities and how fast you're going to be able to get this out to most people in the world. I've already had your vaccine, by the way, and I'm so happy that I did.
This is great, Patrick. Great to hear. So at the end of the day, the great challenge, of course, is to produce sufficient doses. And messenger RNA is a new technology, and there were no established manufacturing capacity. So when we started our program, the usual batch size that we were producing for clinical trials was in the range of 1,000 to 5,000 doses, yeah, which was already a plenty amount to do clinical trials, right? Yeah. Now we need to produce more than 1 billion doses. So this is really a factor of 10 to the 5, 10 to the 6 more than we had produced last year. And of course, this is an extreme engineering challenge, but it is also a challenge with regard to raw materials. So the vaccine is composed of mRNA, so we need nucleotides, we need caps, we need lipids, and the lipids are one of the key limiting components because these are special lipids which are not existing. They need to be synthesized. So we are working with a number of companies who provide us this lipids, and they had also to scale up their production because they usually produce this lipid in 100 gram or 1 kilogram batches, and we need now much larger batch sizes. And that's the limiting factor at the moment. But I'm very confident that with now all the upscaling activities, so we increased the number of vaccines that we plan to deliver. So we plan 1.3 billion doses to deliver in 2021. And together with Pfizer, we figured out how we could increase the manufacturing further. And we are now able to deliver up to 2 billion doses in 2021, which would provide vaccines to 1 billion people. And this is with other companies also coming now in and being also able to deliver vaccines. I'm really confident that we will come in a situation in late summer and that most people will have immunity. Everyone who wants to get a vaccine will receive a vaccine, and I believe that we will get control. We will be able to control this pandemic in late summer, and this is really becoming now more and more concrete.
Well, wonderful. That's really impressive to scale up from 5,000 for clinical trials. And you're right, that's plenty for clinical trials to 2 billion. That's quite a scale up that you had to do. And I didn't realize that it was really the lipid component that was the rate limiting step. So tell me, what's the future hold for RNA vaccines in infectious disease? We're so backwards in how we make a lot of our flu vaccines and other things like that. Do you see that this technology is going to start to take over? And how many different epitopes can you put in there? Do you think you're going to make one day a COVID, flu combination vaccine, for example?
Yes, so the technology has an extremely broad versatility. And so you can combine, so in our cancer trials, we are already combining 4 antigens or 6 antigens delivered simultaneously to the patients. And this is, of course, possible in the same way for infectious disease vaccines. So you can combine several antigens for one virus or combine antigens for different viruses. So this is technically feasible. So the key advantage of mRNA is another key advantage is to be able to make fast a vaccine. So the manufacturing itself takes only less than 2 weeks. Then we have about 2 additional weeks for quality control and sterility testing. So that means you can, in principle, deliver a vaccine from the scratch within 4 to 6 weeks, and this is of course important advantages compared, for example, to the viral vector vaccines or to the recombinant protein vaccines. But another advantage is really that we are able to engineer even better immune responses. So what we have seen in the cancer immunotherapy field is that we can use the same vaccine to prime, boost. For some patients, we have applied the vaccine 30 times, and every time when we apply, we are able to boost the immune response. And that's not possible, for example, with vector-based vaccines, where the vector backbone response is limiting the repeated boosting. So here we have an advantage to have a vaccine which can be used for boosting. And another advantage is that the innate immune response driven by messenger RNA vaccines seems to be strong enough to get immune responses also in elderly people. So the efficacy of our vaccine in people older than 65 years is still the same. It's over 94%. And this is, of course, an important piece. For example, if you would like to use the vaccine for influenza, where we know that the efficacy, particularly in elderly people of influenza vaccines, are not so great. And therefore, there's an opportunity to improve here also for this type of application.
Yeah, I am concerned about the adenovirus-based vaccines because you can only give so many of those before you're going to get neutralizing antibodies, so you really can't limitlessly boost those. But with this one, you could give many different boosters because there shouldn't be any backbone reactivity. Can you tell us a bit about, it does cause some swelling. I actually tolerated mine very well. I had a little muscle aches on the second one. But what's causing that activation of the innate immune system? Because what I understand, it's not really the sting pathway from the RNA component, but it's the lipid component. Do you understand the mechanism of activation of that innate immune system and some of the muscle aches and other probably good effects that we're having from this?
This is indeed an excellent question. So the mRNA that we have used in our vaccine is nucleoside modified, and therefore it should not activate the TLR pathways. Yeah. What drives the immune response is the combination of the lipid component with the vaccine. Yeah. So the lipids are fusogenic lipids, and one hypothesis is really that the disruption of the membrane of the cells initiates an innate immune response cascade. Yeah? And what is driving that is still not known, and something that needs to be investigated deeper, whether the release of intracellular components like ATP or the cell death pathways contribute to that. We have to learn. So it's a matter of ongoing research.
That's really fascinating. Tell us exactly what you think is happening when we get that IM injection of the vaccine. Are the antigens mostly being expressed by the myocytes, the muscle cells, and then being taken up by dendritic cells and going to the lymph node? Or what mechanism do you think is really going on there?
So we have optimized our vaccine to the mRNA LNP vaccine to be delivered to dendritic cells. So the mRNA contains UTR regions which are optimized for strong expression in dendritic cells. And what we see in mice is after IM injection, significant amounts of the mRNA expression happens indeed in dendritic cells and APCs. Yeah, and this is not only the regional draining lymph nodes, but also the distant lymph nodes, which are expressed. And this is key. So for CD8 T cell responses, it's really important to have a direct expression in dendritic cells. Otherwise, you would get just CD4 T cell responses, the exogenous antigen presentation pathway. But we have, with our vaccine, we have shown that in our immunogenicity testing, we have very strong CD8 T cell responses. So the most important is not the expression in the muscle, which is relatively little, but the antigen expression in the draining lymph.
I see, because you probably don't have too many dendritic cells in muscle tissue anyway. So you think it's actually traveling through the lymphatics to the draining lymph node, and then transfecting the dendritic cells in the lymph node itself to then present to both CD4, CD8, and B cells. You think all three with your epitopes? How many antigens do you think you can put into one of these vaccines before you get some issues like immunodominance, where one antigen will dominate over another? I mean, I'm wondering how many vaccines we can make out of one, how many different viruses we can protect ourselves with one vaccine.
Immunodominance is something that will happen on the CD8 level. So you know, CD8 T cells, the cytotoxic ones are egoistic. So they, if you combine a strong antigen with a weak antigen, you get this immunodominance. So that means that it's a question how the vaccine is designed. But if we have an infectious disease vaccine or several antigens in an infectious disease vaccine, then we are, all of them are neoantigens, and we hope we expect that there is some sort of selection of best epitopes from each of the immunogens. With regard to antibody responses, we know that antibody responses could be limited by antibodies binding to the immunogen. It's a negative feedback loop. But again, the advantage of an mRNA is that the mRNA lipid nanoparticle is not the antigen, it is just delivering the antigen. So that means delivering the antigen to the dendritic cells and expressing there allows really that the antigen is hidden, and not just blocked by circulating antibodies. So we have still the opportunity to induce multiple antibody responses in parallel. How many antigens could be used in this setting? I don't know. We didn't test that. I would assume that there is a limiting number because at the end of the day, it's a matter of focus. If you ask the immune system to make immune responses against multiple immunogens in parallel, there will be some sort of reduction on the individual level.
Let's talk a little bit about immune memory. How long do you think people will be immune for after having taken an RNA-based vaccine? Do you know from your cancer studies, can you predict with the COVID vaccine, is it going to be years?
So in principle, we have to ask the question in different ways. So the first question is how long, what is needed to avoid infection at all? Yeah? And the prediction here is to avoid infection, you would need really high neutralizing antibody titers. So neutralizing antibodies are the key to provide infection. If you ask the question how long is protection for avoiding severe disease, this will be much longer. Yeah, because avoiding severe disease can already be happening by having sufficient number of memory cells so that the immune response can kick on relatively early. And even if you get infected, it's not a severe disease because the immune system can catch up and control the virus infection in a few days. And I think this is the way how vaccines work. If the immune response is weak, they can prevent severe disease. If the immune response is very strong, they can prevent infection. And here, at the moment, in the pandemic situation, we are, of course, interested in both. First of all, we would like to prevent infection, because if we can prevent infection, then we will also prevent transmission. Yeah? And but the second best thing that we can do is prevent severe disease and thereby avoid that people die by the infection. The goal is the latter. Then I believe that such vaccines could have a memory effect for years. If we really want to avoid infection, then every year a booster might be useful.
I see. That's really clear, because every viral infection is a race between the viral production and the immune system and the immune response. So what you're saying is that if we want to avoid total infection, even avoid sniffles or a little runny nose, then we may have to have a booster. But if you want to avoid going to the intensive care unit, then maybe that will last years. That's very clear. What's the future hold for these vaccines for infections? Do you think we're going to be able to one day get a flu vaccine against common epitopes that will, so we don't have to predict the strain coming from the east and try to guess on that every year? Or do you think we're going to have a common flu vaccine based on RNA technologies?
I'm not so convinced about the universal flu vaccine. So I believe that a flu vaccine with strong antibody responses could prevent infections also against other flu strains. That should be possible, but I don't think that this will be a universal approach. So influenza has the co-evolution of our immune system with influenza resulted in a virus which is really able to adapt to any kind of existing immune response. And we need to be prepared. If we are smart, the viruses will figure out a way to be able to reinfect. Therefore, I still believe that the best way to deal with that is to be able to respond very quickly to strain changes, and tailor it precisely as we do in oncology. So precision oncology is about really understanding the disease, understanding the molecular nature, and reacting in an appropriate way. And precision vaccines should do the same. I prefer to be tailored as possible.
Yeah, a lot of the conditions that potentially caused the COVID-19 pandemic are still in existence. We're still invading the rainforest, and the animals from there. The rainforests often cause these diseases in man. So how, as a society, a global society, should we best prepare for the next pandemic, whatever that is, whether it's coronavirus or other kind of virus? Do we need to start to survey more, have more lipid and RNA raw materials stored up in the stock room? I mean, what should we do, and how fast will we be able to get the next one out if we're really ready for this?
Yeah, this is a great question. I think this pandemic shows that pandemics could happen. And there were a lot of people warning about exactly the situation. And if you want, you can say that we were still lucky with this pandemic, because this is a deadly virus, but it is still not the worst thing that could happen. Yeah. And the next time we really need to be prepared better, because the likelihood for a pandemic increases because the mobility on our planet increase. That's the most important aspect. Yeah. If something pops up somewhere, the likelihood today is much higher that it will be distributed very fast. Yeah. And what we need to do is we need better surveillance. We need to have production capacity in place, because this was really the most limiting aspect. So I think there were some programs with fast pandemic response, but there was no production capacity. That's really one important aspect. We need dedicated institutions and companies who can just scale up and make billions of vaccines in the shortest possible time. So we need a preparedness. We need a preparedness on the political and international level. Yeah, and we need also to reconsider when we are designating something as pandemic. So if you designate something as pandemic after it became pandemic, it's not the best thing. I think we need to initiate certain levels of response much earlier, before the pandemic is really seen by everyone. But of course, this requires a lot of discussion of experts and politicians, and I hope that we can do that in the next 12 months to establish a program which works in the next outbreak situation.
I think you're absolutely right. COVID-19 is horrible, but it could have been a lot worse if it was a more deadly virus that spread even quicker. It could have been a lot worse, Ugur. So if we do all the things you're saying and really prepare, do you think we could get the next one going for the next pandemic within 3 months or so? You're able to do a personalized vaccine in 4 weeks.
Yeah, I think in principle, the production can be really done faster. So if we are prepared, the key question, of course, is if the pandemic is extremely worse, how to deal with efficacy trials. So this is also something that needs to be discussed early on. Yeah. So we had now the privilege in this pandemic to come up with safety data and efficacy data. It took us, the total trial took us about 5 months, yeah, to do. Yeah. But what happens if we don't have this 5 months? So we need also to discuss that. Of course, the best way is to do this large trials and to prove that you have an efficacious vaccine and safe vaccine. But what happens if you don't have the time? That is also something that we need to discuss.
Yeah, we may have to have a system where if you're making neutralizing antibody in some assay, go. You just have to go and start production. We may have to make those guesses if the next pandemic is even worse, which it very well could be. Tell us where we're going to stand now with cancer vaccines, because I do believe that the lack of specific immune cells against the cancer is a huge rate limiting step. We've had a lot of advances with immunotherapy, but it's mostly cancers that have a lot of immune cells already there that we're then activating with immune checkpoint blockade and others. There are a lot of cancers where we don't even have enough immune cells against the cancer. Where do cancer vaccines stand in your cancer vaccine? I know you're doing a very large study with Genentech and other people. Where does this stand, and will this be the next breakthrough to try to get enough T cells so that we can then utilize immune checkpoint blockade and other things to cause a significant decrease in deaths?
Yes, this is indeed the key question that we have to ask. So we know that cancer immunotherapy is driven by cancer antigen specific T cells, and we know that these T cells are not around in a number of cancer patients and in a number of diseases, and we know that some cancers have little number of neoantigens, and the question is, can we find a way? Can we develop vaccines which enable to induce T cell responses even when there is no spontaneous immune response established? And this should be the core superpower of vaccines. Yeah. So that means a vaccine should be able to develop meaningful immune responses that and induce T cells that can change the disease. And this is what we do with our mRNA vaccines. We developed vaccines that could also allow us to induce strong T cell responses against non-mutated antigens, which is very encouraging. But what is also important to understand that this is a numbers game. Yeah. So we are dealing in advanced cancer with just too many cancer cells. Yeah, and if you calculate the number of immune cells that we are generating, then it is often we are not able to catch up with the growing tumor, and therefore one of our goals is really to go into tumor conditions where the number of tumor cells is limited. That means these are the cancers with minimal residual disease, which might be detectable by tumor markers like circulating tumor DNA or circulating tumor cells. And this will be a focus of future work, where we would like to bring cancer vaccines to this patient population who have a higher risk of relapse, and see if cancer vaccines could enable a better disease control here.
Do you think they have to be against mutated antigens? Because you had some very nice data using differentiation antigens and other cancer testis antigens, if I recall, right, with your cancer vaccines.
No, we believe that also the non-mutated tumor antigens like your ESO or members of the MAGE family, we have expanded T cells, and we have shown that these T cells perfectly recognize and kill tumor cells. Yeah, so there should be no dogma So the utility of the antigen is really based by the ability to induce T cells, which are able to recognize tumor cells and kill tumor cells. So this is the utility question, and everything which is able to accomplish that is a good antigen, and therefore we have clinical studies really exploiting both types of tumor antigens. And of course, we have also the viral neoantigens, which are also excellent targets for cancer immunotherapy.
So, Ugur, you work so hard and you've been so effective, and you've helped the world so much. What do you do to relax? Do you have any fun hobbies?
I love really reading a lot. Yeah, about science, technology. I read the publications of other colleagues. Yeah, I entertain myself by learning and being curious. And of course, the other time is taking time for the family and taking time to do some sports. So I think it's very similar to other colleagues who are dedicated to science and technology.
So now that you're vaccinated, are you traveling again?
Not yet. I really find it effective to use all the communication technology, video conferences. Yeah, I learned that instead of traveling 2 days for a meeting of 2 hours, it is really more effective to have a longer meeting and discuss in more detail. So traveling should only be done if you really combine that with meeting people and spending time with them. One of my goals is, if the pandemic is over, really reconsider how much I'm going to travel.
I think you're absolutely right. I'm going to do the same thing because Zoom is so effective. But one of the most incredible meetings every year is the SITC meeting in November, and you're a SITC member. I'm really hoping that you do come join us in November for the SITC meeting.
I will definitely. This is a special meeting, and it's an opportunity to meet a lot of friends.
Wonderful. Well, that's fantastic to hear that you're coming, and maybe one day I'll get you to visit Moffitt Cancer Center too in Tampa, Florida. I would love to have you down here too. Florida's a fun state, and Tampa's a beautiful city to visit. I've been to Mainz a few times for the KIMT meeting, and I love Mainz. It's a beautiful city on the river, and your university is outstanding as well there.
Thank you so much. It would be a pleasure to come to Tampa, and you are cordially invited also to come to the next KIMT meeting. Yeah.
Well, Ugur, this has been such a pleasure and a thrill to talk to you today, and we're all so proud of you and so encouraged by the vaccine and the results. I mean, it tells us in the end, with the pandemic, it took a cancer researcher and cancer immunologist to solve the issue, and it's the immune system that's going to reverse this pandemic. We're also proud of you and your work, and so thankful and grateful that you've given this vaccine to the world.
Thank you so much, Patrick. But I have also to say that we learned a lot of in the cancer field from virologists and infectious disease immunologists. So it's good that we could give something back.
Yeah, you're absolutely right. Our listeria models and all of the models of viral infection, you're right. We've learned a lot from them. You're absolutely right. Well, Ugur, thanks so much for spending the time today with us. We really appreciate it.
Thank you, Patrick. It was a great pleasure.